Okay guys, welcome to the mandatory practical that goes with the plant responses chapter. It's page 194 of our notes book. There is the QR code there, so you have the choice of either looking at the QR code first or going through this version first. I would recommend you do both. So the QR code is on page 195. You will always see in the questions they will be mentioning the effect of IAA, which is a growth regulator, on plant tissue. So we haven't done this technically yet, but a plant growth regulator does what it says in the tin. Obviously we can't use this word in our answer. So a plant growth regulator is just a chemical that controls how a plant grows. So I'm going to do this possibly slightly backwards, but we'll see where we go. So what you are going to do is you are going to set up what's called a serial dilution, and we'll go into the detail on that in a little while. A serial dilution of your IAA, and this is just a biology way of saying we're going to make solutions with different concentrations. And once we've done that, we are going to take a Petri dish. We are going to put some cotton wool underneath, so I'm not going to draw the cotton wool, and we are going to put a piece of filter paper on top of our cotton wool. So this is going to be our filter paper. We will take a pencil line just so we can, and we will draw a pencil line on our filter paper before we put it in, and then we'll take something like radish or cress seeds and we will line them up here and we will spread them out. Now we put in enough cotton wool so that when we put the lid on top of this dish, that the seeds will be stuck there. They're not going to fall down, and we'll talk about the storage of those in a second. We will also put on top of this an acetate, so that is the see-through plastic stuff, but I will have photocopied graph paper onto it, so I'm gonna have to bear with me while I draw my graph paper. So what this will allow us to do is, once these seeds start to germinate, we will be able to use the graph paper to figure out how much the seeds actually grow. So we'll be able to say, oh, it grew 4 boxes. 4 boxes is 1 centimeter. There's a piece of maths that you won't have to do for the SEC, but we'll just have a look at it. So this is our acetate grid, and that's going to help us with our measurement. So we would now have 8 of these Petri dishes, and we actually stack them like this. So if this is the bench, we will stack the 8 dishes like this, such that, and I'll use the red to do it, what we want is when the roots and shoots germinate, we want the roots to be able to grow straight down, and we want the shoots to grow straight up so that they're easy to measure. If we were to store the plates like this on the bench, so sideways, what you would find then would be they'll follow the whole geotropism and phototropism. So our roots would try grow down into your paper, which wouldn't be ideal. We couldn't measure them, and the shoots would grow up and they'd hit the lid of the Petri dish, and again they'd bend. We wouldn't be able to measure them. So we actually store these plates on their sides so that the roots and shoots will grow up or grow down. Now you have a version of this graph picture on page 195. If you wish to add to it, please do. So in terms of then our experiment details, so we're going to use the different dilutions of IAA. So IAA is probably a version of auxin, and auxin is a growth regulator, and we'll talk more about that in our next lesson. But what we're trying to do here is we're trying to use the different concentrations or the different dilutions of IAA to figure out the effect on root and shoot growth. So when we look at our results for this, we're going to look at the IAA under 3 criteria. We look at it under low concentrations, we will look under it at high concentrations, and we will look under it at very high concentrations of IAA. And that will make more sense when we look at the graph that results. So I will have all this done for you. We dissolve 0.1 of a gram of IAA in a little bit of ethanol, and obviously ethanol is flammable, so I have to be very careful there. So, and we would once it's dissolved in the ethanol, we would add a little tiny bit of water and we would evaporate off the ethanol. Ethanol evaporates at 78 degrees, much lower than water, and then we add it to the 1 litre of distilled water, and now I've made what is called a stock solution. So the technique that I'm going to show you is called a serial dilution. You use it in chemistry for making different concentrations. In biology they will do the same thing because they still use solutions for analytical methods. Even in microbiology you would do serial dilutions. So on the top of page 195 you have a version of this picture, and I'm going to explain what it is that it's going to look like, or we'll make it together, shall we say. So we go out of this and we will go into this. So here we have 8 tubes. In our very first tube we are going to put in 10 mils of our IAA. So this is it's called a stock solution because it's the solution that you're starting with. It's got known concentration and you're going to make everything else from this stock. So it's going to have 100 mg of IAA per litre. So that is a known concentration of stuff, and away we go there. For each of the other tubes we're going to put, book it out, 9 mils of water into each of the other tubes. So every tube here, rest of them, these guys will all have 9 mil in total by the time we are finished. They'll all have 9 mil, but there's 9 mil of water to begin with. And then what we do to do our serial dilution is we take 1 mil and we put it in here. We would give it a little shake, so it has 10 mils now, and then we remove 1 mil of this and we put it into here and we give it a little shake, and we will then do the same thing all the way down. So we take 1 mil from the preceding tube and we put it in to the next tube, and what we are doing is for every time we do that we are diluting it tenfold. So this first tube was 100, this next one would be 10 mg per litre, this one will be 1 mg per litre, and it'll decrease tenfold every time you do this. So you, as I say, use it in chemistry and biology to make standard curves, they're called. So you would use it as an analytical method, and you also use it in microbiology with bacteria. So you'd have your stock culture of bacteria. There might be thousands upon thousands upon thousands upon thousands upon thousands of bacterial cells in there, and you would serially dilute and you'd plate all of those cells, and then you'd finally get a plate that you could actually physically count the cells in there. So it wouldn't just be one big smooth colony, you'd actually have little individual cells finally. So that's a micro use. So this as a method, once you go to university, is a thing. So for this very last one, there's no other tube to put something into at the end, so you remove 1 mil and you just toss it down the sink. So then every tube here, they will all have the same volume, they are all different concentrations, and then we just label our Petri dishes. So if this is dish 1, this is 2, 3, 4, 5, 6, 7, 8, we would label our Petri dishes in the exact same manner. So when I'm done, I'd have 8 Petri dishes, and this might be dish number 2. Okay, so that would be how we would do our serial dilution. So going back to here, it just says we started with 10 ml of IAA, we diluted 1 in 10 7 times, and we're going to put this word in serially. So if they ask you how do you do your different dilutions, it is a serial dilution because you're taking one and you're doing it in series. And obviously then at the very end we also have a control plate, and our control is just water. There's no IAA in our control. Okay, so in terms of the types of seeds we're going to use, or you can use, we use radish or cress, and we do that primarily because they're really fast to germinate. So it'll only take a few days before we can see a result, so that's always good, and they won't grow so fast that they'll grow out of our Petri dish. We add equal amounts of each dilution and control because we want the volumes to remain the same. the only difference should be the concentration of IAA. So this is our the volume is a control variable. So we want to keep the concentration of IAA. That's the only difference that we're going to see. And the control we've just said is just distilled water. So it's for us, we make sure if 9 ml is our total volume for everything, then it'll be 9 ml of water. And we've already explained this. So we stand each petri dish vertically so that the roots will grow straight down and the shoots will grow straight up. So if you want to just put your little quick picture here, your line, these are your seeds, you'll have more colors than me. You want your shoots up and your roots down. So that's what we mean when we say the dish is vertically. We will incubate them for 2 to 3 days. We actually leave them at room temperature, and we want to make sure that the seeds have germinated, and we want to make sure that the roots and shoots are long enough so that we can measure them if we so wish. I should have said that we take 5 or 6 seeds, and we do that to ensure that some of them, at least if only some germinate, we'll still get a result. So some will at least germinate. So I apologize for that oversight. Okay. How will we measure the length of the roots and shoot seedlings? Well, we use that acetate grid. By including that, we can use that as our measurement. And there is a piece of maths that we use. Now, the SEC have never asked it, but technically they can. So we do work out a percentage. And what it is we take so this might be for, we would do this for each plate. So if we took it for plate 2, it would be the average length of a root or a shoot in plate 2 minus the average length of the control, multiplied by 100 divided by the average length of your control. And you'll either get a positive answer or you'll get a negative answer. And if you get a positive answer, that is what's called stimulation, because your root or shoot has grown. And if you get a negative answer, that is what's called inhibition. Now, whether or not the SEC would ever ask this as a calculation, as in just vomit out the formula, they haven't asked it yet. So we will see. The most important thing is, so just to reiterate, if you have positive growth, then that is stimulation, and if you have negative growth, that is inhibition. So please make sure you've written your formula and that you have positive growth is stimulation, that's how you refer to it, and negative growth would be inhibition. So this is our graph, and I'm going to try and see, can I do different colors here to explain this. So we're going to start with this one. So when you look at this graph, there is 2 lines in here. The red line is the root line and the blue line is the shoot line. And earlier I had said that we're going to look at the result in terms of very low concentration, high concentration, and very high concentration. So this is going to be our very low concentration. And what are the graph telling you in terms of root and shoot growth? So at very low concentrations, so this is our stimulation, so this is our positive growth up here if we got a positive answer, and down here is if we had a negative answer in our maths. So at low concentrations, the information that we want is that roots are stimulated and shoots are inhibited. So that's the first observation. So that's this little green area. Now there's the blue line here, I will grant you, is slightly increasing at the bottom, but for the most part, low oxygen concentration stimulates roots, inhibits shoots. And the reason for that is that initially when a seed is germinating, it needs to grow roots to get water to help everything else happen. So roots will always develop before shoots. So in terms of our next area then, we look at high concentrations. So for high concentrations, we are going to go with roughly to about here. So if you want to color this in a certain color, you can. So this is going to be our high concentration. Match our pen. So high concentration, you can see the way the graph is working. If you have a high concentration, it is now roots are inhibited, but shoots are stimulated. So when the concentration of auxin gets so high, your roots stop being stimulated to grow. They just don't grow anymore. They don't get any more bigger, but your shoots are. And if we go to the last one, which is very high concentrations of auxin, you will see here that the line is now starting to go down for the shoots as well. So in terms of then our explanation for that is at very high concentrations of IAA, both roots and shoots are inhibited. So we have 3 categories of IAA, and we talk about the results in terms of roots and shoots for each, and if the line is above this little gradated bit, then that is stimulation, and if it's below, it's inhibition. And this is our auxin concentration down the bottom. And obviously we're starting low. It's low over here and it's high up here. At the bottom of page 196 you have explanation of this experiment in terms of its procedure again. And that is the end of our